So it'll be interesting to see because this is the first time we've done one in since school's out, and so you're at a research institute, so I'm not sure if you noticed the difference between school being in or out.
Not really.
Yeah, but I mean here campus is dead. So you did your PhD at Penn State with Lynch, but then didn't you have some connection with Benfey too at some point?
Not Phil Benfey. I went to Nottingham for about 2 years at the University of Nottingham with Milton Bennett.
Oh, it's your postdoc?
Yeah.
Oh, okay.
And I had a brief postdoc on Felix Ritchie at the University of Missouri before I joined Nobel.
Oh, that's right. Oh, there's your CV. Okay.
You find one somewhere?
Yeah, I found one on your Nobel site.
Okay.
So you are like the most active social media person I can think of. How do you feel? Do you feel like that sort of helped your? I mean, that visibility is part of the reason I thought you'd be a good candidate here, because I can tell you're obviously quite engaged and active. But
it helped in that sense, I guess.
Yeah, but anything else? I'm sort of curious about this because we're entering into a new age, and like what type of things people need to do to sort of promote themselves and their research and like help students get further their careers.
Yeah. I think that showing your research is really helpful. I'm not sure, like when they interviewed me at Nobel, whether people knew much about my web presence. I always maintained a website with information about my research might be available. And to me, it's like if you're not discoverable by Google, then you pretty much don't exist. So
that's pretty funny.
I mean,
if you're not discoverable by Google, you pretty much don't exist.
Professor, and I looked them up. I can't find any information about them. That's not very helpful. So.
Huh. And I forgot. I saw you sent out a tweet on your seminar, but really, when pollination happens here, I lose my mind. So I feel like I'm a little bit out of touch with reality.
Okay. I thought maybe you might be a little Twitter-pated. I didn't know if it was you doing the pollinating for the plants or what.
Well, you know how we pollinate corn, right?
Well, you have to bag the tassels and I guess shake the pollen onto the silk, I guess.
Oh yeah. But more importantly than that, I mean, it's all about setting the mood. So you got to turn the lights down low, and you got to put the berry white on. You got to get a little glass of wine for each of the corn plants, and
that gets expensive, huh?
Yeah, and really time consuming too. But no, my group makes like about 30,000 pollinations or something. So that takes about all day, every day for about 3 weeks.
Wow, that's a lot.
But we're not talking about me. We're talking about your research. So this is kind of a thinner crowd than I'm used to. Hopefully that's not because I didn't tweet it out, or you didn't tweet. Well, you did tweet it out. But what I usually find is that a lot of people will join here in over the next 5-10 minutes too. And I'll send a couple reminders once you get going to other groups. But did you get my reminder this morning or last night?
I think you sent me one directly. I don't think I saw one that was part of a group or anything.
Okay. Oh well, it is 11. So and just so you're aware, Larry, when you have things on your screen, like you have some box that's popped up, it's grayed out so we can't see it, but it puts a gray thing right over the slide. Same thing with, I don't know if that's the WebEx thing that's hanging down on top of your functional phenomics, the very top of the slide.
Yeah, it has this, you are sharing.
Yeah, okay. Normally that doesn't show as grayed out for us, but. Well, I guess it's probably time to get started here. So let me find my little script. So it's my pleasure to welcome to, I think this is our 8th Plant Phenome Journal webinars in the series for the month of June. And so today it's Dr. Larry York, who I'm very excited about getting to see his research and getting to hear about what he's up to. He's an assistant professor at the Nobel Research Institute, and he focuses on root phenomics. So Larry actually, or Dr. York, got his bachelor's at the University of Kentucky. He did his PhD at Penn State University and with Jonathan Lynch. He was a postdoc both at the University of Nottingham and at the University of Missouri with Felix Fritchie. And then he joined Nobel in 2017, where he's been very active in developing some new phenomics methods and is also a very, very active social media user. And that sort of grabbed my attention with all the updates he gives on his various research throughout the week. So I knew he'd have plenty to present on. So Larry's going to be talking about functional phenomics, an emerging field integrating high-throughput phenotyping, physiology, and bioinformatics. And Larry, go ahead and get started. And I'm going to mute people as they join, so hopefully you don't get any feedback.
Okay. Thanks a lot, Seth. Thanks for inviting me to do this webinar. I hope I'm not oversharing on social media, but I'm excited about research, and I like to show my work to anybody who wants to look. So I do enjoy that aspect, and I think it's helpful for people's careers to be visible. Today, like you said, I'll talk about functional phenomics, and this is kind of a rebaking of my normal talk to highlight what I think is an emerging field and the topic of a review paper I just had accepted with revision to the Journal of Experimental Botany with the same title. So I'm going to talk about that and how it relates to my own work. Before I go into the research, I just wanted to show a slide showing the campus of the Nobel Research Institute in Ardmore, Oklahoma. We are a nonprofit agricultural research organization that's focused on trying to solve great challenges that agricultural producers face in the southern Great Plains. And if you can see my mouse where I'm circling is actually the windows of my lab in that building. So let's start with what is functional phenomics. Basically, I think that high-throughput phenotyping is the central pillar of functional phenomics. And what I mean is that if we can phenotype things like links, angles, and numbers of roots, that we can learn more about how plants operate. Specifically, functional phenomics is the quantitative study of phenotypes and their relations to performance, whether it's agronomic in the agricultural setting or fitness in natural systems. I really think that phenotyping and phenomics also applies to natural systems, but really haven't been applied to the degree that they deserve to be. So, for example, if we were to measure the root properties on many different plants, and we also measure the shoot properties like mass, yield, nitrogen content, etc., we can link the variation in root properties to the variation in shoot properties and maybe be able to infer something about how plants work. So phenomics lacks the vocabulary to adequately conceptualize the relation of phenotype to function. And so here is a proposed one-to-one mapping of phonetic and genetic terms, going from phenome, genome, phenotype to genotype, phene to gene, and phene state to allele. The word phene itself has been used almost for 100 years, and some people believe it's implicit in the same work that coined the terms phenotype, genotype, and gene. One important distinction of a phene compared to a trait is that it's a property of an organism, whereas trait is often used across biological scales. And another important distinction, a big problem in genetics and in phenomics has been the interchanging of words, for example, like gene and allele. A lot of times people say gene when they mean allele. And similarly, when we talk about phenes, people often talk about the general property in place of the specific attribute. And a good example of that is that people might say things like the eye color phenotype. They may also say the blue eye color phenotype. So they're calling both the general and the specific by the same term. So this terminology is trying to get people to think about the differences and be more clear in what they're saying. So another thing I've proposed in this paper is what's actively been guiding my research since I started at Nobel, the functional phenomics pipeline. And you can really start anywhere along what I'm going to show, but I think that generally the most useful place to start is with deciding what your concept of an IDEO type is for the plant that you're working with and the environment that it's growing in. So an IDEO type is essentially an ideal phenotype that will maximize agronomic production or profit and or fitness in natural systems. And there's been a lot of debate in breeding about the difference of selecting for yield, which is what most breeding has been doing for hundreds of years, versus idiotype breeding, where you actually say, I want maize plants with erect leaves, with steep roots, and all these traits, and we want to stack them together. So the idiotype is a good place to start because it basically makes you be explicit about what you're looking for. And only after you know what you're looking for can you design a phenotyping platform. Phenotyping platforms are basically designed to be as high throughput as possible and as cheap as possible. And for the example of roots, we might have decided that our idiotype had certain attributes like numbers, lengths, and angles. And so our phenotyping platform will be designed to measure those as quickly as possible on as many lines as possible. At that point, you can try to phenotype a population. And since this is functional phenomics, we're not just measuring for the sake of measuring stuff, which is what I think a lot of phenotyping has been doing, just measuring many things and mapping it and then kind of ending the story there. What we're trying to do is make a functional relationship between various phenes and their co-correlations, their relations to performance. So if we phenotype the root properties and shoot properties, for example, of an entire mapping population, which has a lot of diversity that will allow us to make these connections, then that's a good way to understand how plants function. So of course we get things like the phenotypic variation for different phenes, and usually the endpoint a lot of times has been simply identifying genetic regions and then stopping there. And so what I'm proposing, I'm not saying I made up the ideas here, but I'm just trying to make it more concrete and show that it's a real pipeline that can guide our research. So rather than going only to genetic regions, we can also correlate phenes to utilities. And this is where the bioinformatics side comes in. I've realized with my work that many times I have more than 25 root phenes that I've measured, and possibly as many shoot properties. And so we have this complex scenario of these big data sets of variables that may co-correlate and have different types of relationships. It's multivariate. So we really need to push our statistical abilities to infer these relationships. So I've been using things like multiple regression analysis, principal component analysis, and I think there's a lot of room here to make big advances in the future. Another thing that we can do is detailed physiological studies. So I'll give an example of this from our lab's work. Trying to understand, we for example may have uncovered variation in some root phene, and we have inferred that it may be related to performance, but we want to understand how it operates more in depth. So we could, for example, do more detailed greenhouse experiments where we can measure the soil properties over time, measure the roots over time, things like that to get a more deeper understanding. And then last, we can combine this all with simulation modeling. And I also will give an example of how this has played a role in my work, and I hope it can play a role in the future. In fact, I'm going to attend the AppSim training at Iowa State University next week so that I can start using crop systems models in my work. So the simulation modeling is useful because it puts your theory into a concrete system. It makes everything explicit about your theory and allows you to test your ideas in more different ways than only empirical experiments would allow. So that's the functional phenomics pipeline, and now I want to show a little bit about the concept of idiotype and how I think of root idiotypes. So I'm basically going to give you a crash course in root biology. So if you're in soil with roots, you might have an immobile nutrient like phosphorus, shown here in red, and it absorbs and desorbs from soil particles in such a way that the diffusion of things like phosphate are limited. And what this means is that the plant can scavenge and take some phosphate up, but the depletion zone around the roots is going to be very small because of that lack of mobility. On the other hand, if you have something like nitrate that's very mobile in soil, it doesn't absorb as strongly to soil particles, then as the nitrate moves with mass flow with water to the roots and is taken up, you can see that the area of depletion is much larger than it was for phosphate. So those are the basics of soil, and now I want you to consider, what does a root system look like? So I'll give the audience a moment to picture a root system. And hopefully everybody did better than this, but I think a lot of times roots are just kind of viewed as this big mass of noodles, you know, just messy and has no structure, and is hard to untangle. There is more to it than that, but noodles are still a good analogy for roots. So for example, all plants that grow from a seed start with a primary root, or the taproot, that emerges from the seed. And over time, lateral roots will emerge from that primary root. And then if we look at the diversity within a crop species, we might see that some plants have few lateral roots, like you see on the left, and some plants have many lateral roots, as you see on the right. Now, let's combine these ideas with what we know about soil nutrient availability. For example, if you have something like phosphate that's immobile in soil, the depletion zones around the roots might look like this for the few lateral root system. For the system with many lateral roots, you can see that these extra roots are able to increase the area of phosphate depletion, and thus you might hypothesize that more lateral roots can be beneficial for phosphorus uptake. On the other hand, if you were to consider the mobile nutrients like nitrate, you can see that the system with few lateral roots already takes up a pretty big area of the nitrate because it's more mobile. Whereas when you've doubled the number of lateral roots, you haven't really influenced the area of nitrate uptake. And this is important because, like noodles, roots have a cost. They have both construction costs in terms of carbon, and also the cost of maintenance, respiration. And so we know that roots take up a large proportion of photosynthate. So in root biology, in terms of breeding, we've been thinking in terms of having cheap root systems where you only have as many roots as necessary for either taking up nutrients or also, of course, for anchorage of the plant. We can also think about root angles. So, for example, you might have basal roots, and for example, a bean that are shallow and grow into this area of red that you could think of as the layer of phosphate that generally forms in the topsoil because it's immobile and being applied as fertilizer. So shallow roots might be beneficial for phosphate uptake. And then in the case of steep roots, well, maybe they're beneficial for things like water as it moves deeper in the season, or for nitrate that leaches with it. So these are the type of background ideas about how plants work that serve as the basis for a lot of the root work that I'll talk about. So how can we use roots to increase productivity? Well, first we have to identify root themes, and then we have to validate their utility, and then we can breed for roots, which is the ultimate goal that I have, is to serve as a translational between basic science to applied science of breeding and agronomy. So like I said, we started with the concept of idiotype, which shifts focus now to development of phenotyping platforms. So phenotyping platforms prioritize your target themes, and they attempt to maximize throughput given material cost and labor, where labor is often, for me, the limiting factor. Specifically, I'm going to talk about root crown phenotyping, or shovelomics, as it's popularly called. And this is a method for high throughput measurements of the excavated top portions of root systems from field-grown plants. And I've recently released a chapter in a root development methods and protocols book about phenotyping in maize, wheat, and soybean that gives some good advice. The first experiment I'm going to talk to you about is from the Ukulima farm in South Africa. This was a farm that was funded by the Howard G. Buffett Foundation. So in order to understand roots, you have to know something about the root system. So the structure of the root system in maize is having seminal roots, such as the primary root that emerges from the seed, and then these other seminal roots that emerge from the scutellar node. And then the stem grows up from the seed, and over time, crown roots or nodal roots emerge. And eventually, you've probably seen in maize that you'll have these nodal roots that form above ground that people call brace roots, although it's not really clear that they actually help to brace the plant. So what I did when I started at Penn State was people were already measuring root crowns there, but they were doing it manually, and they were only looking at sort of the outside of the root system. So this is a root crown that was excavated from the field. It's about 20 centimeters deep, and so we're definitely losing a lot of roots with root crown phenotyping. It's not the whole root system, but we sort of view it as the backbone of the root system, and it does provide a lot of useful information. So what I wanted to do was to phenotype the root system more intensively. So I imaged the roots just using normal cameras, trying to take high contrast pictures. And I measured things like the angles and the numbers, diameters, lengths, as you can see. And over time, I cut away the roots from a plant, which basically shows the development. As we cut away roots, we reveal these older layers of nodal roots that are thinner. So these are actually the older parts of the root system. So we're sort of going back in time as we cut away roots. And finally, we get to, this is the first whorl of nodal roots, and this is the seed. You can see the remnants of the seed and the primary root and seminal roots there. So I measured the numbers and angles, lengths, etc., from all these different whorls. So then that goes from phenotyping to looking at the variation. So this work hadn't reached sort of like my target now of a whole population. It was only 10 genotypes total. And what I'm showing you here is just 3 genotypes that varied for each root theme. So this type of detailed analysis allowed us to see that there was variation along the node position, where 1 is the older roots and 7 are the newest roots. And we can see this is the nodal occupancy, so the number of roots per whorl. And we can see that this increased as the plant developed. It produces more and more roots. And we can also see that in general, there's a difference between plants that were grown in blue being high nitrogen and red being low nitrogen. Angle also varies among the node positions. So for example, it tended to go from more shallow to more steep as we move up here. So these are shallow roots and steep roots. And in some cases, it flattened off to stay steep as they emerge. In other cases, that they might even become more shallow again. So it was clear to me from this work that only measuring, I need to close some windows here. It was clear to me from this work that only measuring the outside roots could be problematic. So another theme we looked at was the distance to branching, which is the distance from where the root emerges from the stem to where the first lateral roots come out. And again, there was a lot of variation among genotypes and node position. So a lot of work focuses on this type of variation of themes, but we wanted to make some more connections among the data. So for example, I was able to show that the number of root nodes correlates to the nodal root number. And the nodal root number, we think, is a sort of governing theme that determines a lot of the backbone that all the other themes are interacting within. And so the number of nodes is much easier to score than the total nodal root number. So it might be a faster way to make selections if you're wanting to decrease or increase nodal root number. We can use principal component analysis. So here I ran the analysis on all the data, and then in the biplot I've color coded by the node position. So you can see the first principal component is largely composed of size-related properties like the diameter, length, number of roots, so that they are sort of sized. And the node is colored here, and you can see that this component can basically predict the differences among nodes. And the second component was composed of angle and branching density. And I've kind of seen across different species like wheat and soybean as well, that this is a pretty common structure where the first component will be these size-related themes, and angle is usually loaded onto the second component. So we're going from just looking at variation now to correlating themes to utility, which is, to me, probably the most important aspect to increase our understanding of how plants work so we can actually make decisions about breeding. So in this case, we have on all these four graphs, we have shoot mass on the y-axis, and then we have different root themes on the x-axis. And so in this case, it's lateral root branching density, nodal root growth angle, lateral root length of the fourth whorl, and lateral root length of the fifth whorl. These were the most strongly related to nitrogen uptake and the high nitrogen plots. But this is all univariate analysis. So to do the multivariate analysis, we need to combine all this data and do multiple regression. And then what happens is that we can actually get a model that explains more of the variation. And in fact, it may allow you to explore the interaction terms among themes and other concepts. So this is an area that I think we really need to make more advances in order to make gains. So now I want to show you a little bit about how we use simulation modeling in this work. You can see I've skipped over the detailed physiological studies, but we'll go back there. And so it's obvious that this isn't just a linear pipeline that you can move around within. So at Penn State, a simulation model called SimRoot was developed many years ago. It lives on now as Open SimRoot. as a collaboration between Penn State, University of Nottingham, and the Forschungszentrum in Jülich, Germany. Hope I said that right. So the way that StemRoot works is that you're actually being explicit. You're planting a seed and growing roots of a plant. It has a soil. So here you can see nitrate leaching down as the season advances due to precipitation. You can see the roots have grown, and then you can see the depletion of nitrate around the roots in blue. So what StemRoot allows us to do is to vary the root genes however we want. So for example, in my work, I varied having few nodal roots and many nodal roots. And in fact, I can have a continuous gradient in the number of nodal roots. And you can see the few nodal roots grow over time. And then you'll see that they have stopped growing, whereas the many nodal roots will keep on emerging over time. And the way that these are color-coded is that the more red it is, the more closely that root segment is located next to another root segment. So it's clear in the case of many nodal roots that you have a lot of roots in the same area of soil that are competing for the same nutrients as other roots. So we think that this can actually be a problem and possibly a poor carbon investment for the plant, which I'll prove to you in a minute. So in the simulation, generally I could see that as I increase nodal root number even from 0 to going up, that in low nitrogen soil, the shoot dry weight was higher with the fewer nodal roots. So it was obvious from the model was predicting that basically when only nitrate is limiting the plant's growth, the nodal root number, having more nodal roots actually is not beneficial. So I think this contradicts a lot of people's basic understanding of roots, is that more roots is always better. Well, clearly it's not. So in this case, we can see a plant with 46 nodal roots and 6 nodal roots. And actually the plant with only 6 nodal roots almost looks like it has a bigger root system. But I promise if you actually count these roots emerging from here, there's only 6 here and 46 there. But because each root here is able to get more photosynthate, then each one can grow deeper. So you can see that the 6 nodal root system is deeper. And you can also see that the lateral roots have been able to maximize their growth rates and are longer than in the 46 nodal root system. So that told us a bit about how roots might work, but then we want to know if it's really true. You know, is fewer roots actually better? So how can we test this? So we can do a detailed physiological study, and here at Nobel last year, a postdoc, Dr. Haichal in my group, did a maze experiment in the greenhouse in these large mesocosms that are 5 feet tall and 6 inches in diameter, with a sand vermiculite perlite mixture inside. And we started with a genotype, and we had high and low nitrogen solution being added to these to create a nitrogen stress. And we manipulated the root system. So this is a direct manipulative study which hadn't been done previously to look at the influence of nodal root number on nitrogen uptake and plant growth in maize. So we had the B73 line, which is a common parent used in maize breeding. We had high and low nitrogen soil levels or media levels, and we had cutting levels of 0% of the roots cut, 33% of the roots cut, and 67% of the roots cut away. So we cut roots away every couple days, checking to see if any root buds had emerged and excise them. Pretty simple. What that looks like, you can see here, when the plant's very young, you can brush the media away and cut the roots. Then you might have these short root fragments. Here's how you can cut them. And basically over time, we were just targeting those 3 cutting levels. And in the zero cutting level, we still brushed away the media and counted the roots. So we have the data for root emergence over time, for example. And at the end of the experiment, at 40 days of growth, we pulled out these plastic sleeves with all the media in it and cut them open and then washed away the media from the bottom up. And then we took the whole root system and laid it out on a table, and we actually separated all the different whorls of nodal roots, and you can see the seminal roots here and the primary roots. So it should be clear that the primary root is much deeper and that the last whorls of crown roots that are emerging are much shorter. And these were cut into 30 centimeter segments so we could get root length density with depth. And so total root length had some interesting results with a clear trend and a significant difference of increasing total root length of each plant based on the cutting level. So you have to remember that the zero cutting level versus 67% of the roots cut away, that this plant, the 0% plant, has 3 times the number of nodal roots, and yet it has about half the length, total root length of the plant. So cutting away roots actually led to a longer in total root system, which I'm not sure if that would be what people would expect, but we think this happens because carbon is being allocated to the root tips, but still they can't meet the demand of the whole root system, and that may free up carbon for more shoot growth that then can fuel more root growth. And so it's sort of a synergistic feedback that allows much bigger, longer root systems. And one way that we can show that this happened is by comparing when we scan these on a flatbed scanner in WinRhizo, we set the diameter classes so that we could tell the lateral roots from the axial roots based on their diameter. And we did the ratio of laterals per axial root length. And so what you can see is that as we cut away the roots, that this more than doubled. So the laterals became much longer, and that's where a lot of the extra length is being accounted for. So the root data with depth is shown here for total root length per depth, high nitrogen and low nitrogen. Pink is 0 cutting level, green is 33% cutting level, and blue is 67%. And what you can see is that, especially in low nitrogen soil, that the plants with fewer nodal roots were able to actually place more roots at depth. And you can also see that even in high nitrogen. And in this experiment, we did also inject the stable isotope 15N with depth. And so we'll be able, and we have that data now back from, I guess it was Davis, I think, did the analysis. And so we'll be looking at, did those deep roots actually, were they able to get that deeper nitrogen source? And then, like I said, we have all this data for every root class. And so we can actually look at the differences among the root classes, which I won't go into here because it's too complex for a presentation. Interestingly, in this study, under nitrogen stress in the low end, the 33% and 67% cutting levels actually increased shoot dry mass by about 50%, and these are significant. And even in high nitrogen, there's a significant trend for greater shoot mass. So these simulation results predated the physiological work, but based on the strong results from the simulation, we conducted these physiology experiments, and we believe that we've largely confirmed the ideas that fewer nodal roots can be beneficial for nitrogen uptake in some cases. So now we'll show a little bit about the work in wheat that I started in Nottingham, and I've continued here at Nobel. So the wheat root system is a bit different from maize. It has a seminal root system, and it also does have a primary root, but here's an example of roots as noodles, where even a root expert has sort of lumped in the primary root with all the other seminal roots, because in wheat, it's really hard to tell them apart. But in wheat development, you have the seminal root system, and then you have the nodal roots that emerge from the main shoot. And then you also have tillering, which actually tillers come off from root nodes, and then they'll have their own root, and then they can produce their own nodal root system as well. So another way to look at it is this branching diagram that shows the main shoot with the different positions of nodes on them, and tillering is shown, and then the different locations where roots can emerge, and then each tiller can also tiller. So the wheat shoot and root system can become very complex and bushy. So when I dug up the wheat root systems, this is what I saw. And similarly to maize, I wanted to look at it more in depth. So I counted the tillers and the number of roots, their angles, lengths, for the whole root crown with all the shoots. But then I also separated off all the shoots, and so then I imaged just the main root system and the root systems of each tiller, however many tillers they had. So we can see in this data for these various root themes and shoot number that you might have one line you can depict like this. So you can see how the different themes vary for this line. And then we can fill it in with all the other lines. In this case, it was a mapping population. And then we have the two parents, Rialto and Savannah here. So you can see that they tend to be in the middle, and there's transgressive segregation of the root properties in a lot of the cases. And we can look at correlations among the root themes. So this is a ribbon diagram where, for example, the crown root length, the wider the ribbon that connects it to another theme, the stronger the correlation. So you can see crown length is strongly related to root number, but it's not really related to roots per shoot. And again, we can fill this in for all the themes and see how all of them correlate to one another. Another way to show this is, of course, with a heat map. One thing we figured out in this work is that root crown width, which is often called plate spread in the literature, and has been linked to lodging resistance in wheat, being that the wider the root crown or plate, the more lodging resistant line might be. But using this approach, we were able to basically dissect this one trait into several other themes. So for example, tiller number relates to root crown width, root growth angle, where more shallow roots, of course, are wider root crowns. The total root number positively correlates with root crown width. And then again, if we do a multiple regression model with all three of these root themes, we can do a much better job at predicting root crown width. And so this theme-based approach is useful at times because I think it can get you to think more in depth about whether a certain property you're measuring might actually decompose into more fundamental units. And I think this is useful for breeding because it should be easier to select for more highly resolved themes. And I also wanted to ask the question when I did this work of, do the root properties of the main shoot correlate to the root properties of the different tillers? In other words, do they all sort of have a common genetic background, and do they develop in a similar way? I think that in this study there is evidence that they do, and you can see this because on the y-axis on these panels we have the whole crown root angle correlated to the root angle of the main shoot, the first tiller, the second tiller, and the third tiller. And in all cases, they correlate, and they all correlate to each other. So it seems to show that the different shoots of the plant develop in a similar way. And so I think that even gives a stronger evidence of the genetic basis for these phenes. But now, a lot of my image-based work has been limited in the sense that it's very hard to acquire high contrast images where it's easy to distinguish the root crown from the background. So even though I was trying to be careful when I took these images, actually if I run them through some type of automated pipeline, it may not be able to properly segment most of the images. So it's just not really an optimized phenotyping system. So when I got to Nobel, I decided that for me to really push myself and my work and be competitive, that I needed to have state-of-the-art phenotyping systems that would allow me to measure properties across whole populations, as I proposed to do in the functional phenomics pipeline. So one of the ideas I had was, why not use these sort of machine vision cameras rather than consumer cameras? These are cameras that are designed for use in factories a lot, to use in automation lines, and they're powered and controlled over USB, so only one cable per camera. These cameras are only about $600, and in this case, it is a monochrome camera, and monochrome can be more sensitive at times than color cameras. So I had this idea that we could use this monochrome camera and get better pictures. And so I built this aluminum structure with a LED light panel in the back. And this is basically the first iteration of it and the first picture that I acquired. And when I got the picture, it looked like this. This is the raw image from the camera. It's not segmented. This is actually what the camera has output. And as you can see, it's very clear. It's very easy to distinguish the root from the background. So this was a success, and then we built on that to design this open hardware platform that we're working on the publication now. So we call it RisoVision Crown, and it's fairly cheap to reproduce. You just need this aluminum structure, an LED light panel, the camera and lens, and these plastic panels. And the total cost is about $1,200 to build one of these. And the hardware is combined with new software. My first hire in the Root Phenomics lab was a computer vision specialist who's done a really good job at helping us make ready-to-use software. We're planning to release this software. Currently, what we're focusing on just the Windows version of it, but it's designed to be easy to use. It doesn't have any dependencies. There's no MATLAB scripts behind it. It's just a standalone, ready-to-use software. And in this case, the Imager software allows you to control multiple cameras. You can modify the acquisition settings of the cameras. It has a live view. And finally, one of the innovative things here is that we actually use a barcode scanner to trigger image acquisition. So we put barcodes on our samples, and when we take the image, we scan the barcode, and then the plot information from the barcode is used for the file name of the image. And so it really streamlines the whole process. So we tried to think it through. Along with this, we have the Analyzer software. And it can output 25 features as a CSV file, and has an option for saving feature images depicting some of the measures. And you can see here, you can choose the input and output locations, and you can see the progress bar as it goes through the images. It can process about one image every 3 seconds or so. And it has an option as well. You can see here, save processed images that will output these images that have a lot of the phenes that we've measured overlaid on top of it. So to give you an overview of this type of image analysis, I'll show you the raw image from the camera here. And then next, we segment that image. It doesn't look much different, but the original image was in grayscale, and now it's purely black and white. So we're able to get a lot of fine details and have no segmentation issues, and it's a simple threshold. We don't have to use any fancy algorithms to do this. We can calculate things like the convex hull, the distance map. Once you have a distance map, you can fit the medial axis to get the skeleton, which you can use for measuring root length. And we can also get the perimeter of all the roots. And we're also using disconnected components to count the number of holes within the root system, which is related to how branchy it is. So these are just a few of the 25, but the ones that are easiest to depict graphically. You can zoom in for a little detail to see even these really small holes. We built a 5-camera version of this last summer. You can see here that it has the clip that holds the root and this holder at the top. This is another innovation that makes this fast, that you have this holder that you just pull out and replace the root and put it back. It standardizes the location. You can see a laptop here with the barcode scanner. In this case, it's 5 cameras along a 90-degree arc, and a simple just one cable per camera, USB hub, and that's all that's required. So this 5-camera version was used in Missouri last summer. They got about 28,000 images. There were no hardware issues, and this was used in the field. So it's robust enough to actually use in the field where you're sampling the roots. We were able to transfer the images to Nobel and do all the image analysis in just a few hours. Well, in this case, probably would take over a day, but still, it's much faster than previous techniques like doing it manually would take months. This is from technically a different system. It's still a 5-camera system that Fritchie and Czar had developed with Anand Sithapalli before I got to Missouri. And when I got to Missouri, I helped them build a final version of it and get sort of the ground truth data. And we have this submitted now and available as a preprint if you're interested. So what we were able to show is that these are all the image-based genes here, and they have pretty good heritabilities over 0.5 in a lot of cases, not in every case. And these are manual measurements that were done, and you can see that the image-based ones have similar heritabilities, but there's many more of them. So we think that these measures could be used in breeding, and we've also already found some other use cases for the system. For example, some collaborators here at Nobel, Carolyn Young and Chox Matupalli, are looking at cotton root rot in the field. You can see the disease front there, a survivor, and the asymptomatic area of the field. And so they sampled roots from along this gradient, and we used Rhizovision crown to measure them. And what you can see above here is the asymptomatic zone roots shown like this, and some survivor zone roots shown here. It's pretty clear there's no taproot in these, so likely the taproot is killed by the cotton root rot disease, and then other roots may emerge to take the place of the taproot. And so, the sort of question is there something unique about the survivors that allowed them to survive, and does it have to do with the roots? And here, these are all the features from Rhizovision crown, and the colors show the deeper blue or red show a bigger change from top to bottom. So basically, this shows that this system can distinguish the different types of roots based on the disease status. And so with that, I want to acknowledge some of my collaborators. and support from the Samuel Roberts Nobel Foundation, which is the manager of the Nobel Research Institute, and provides my base level lab funding that a lot of this work was showing today. So I hope I've shown that the functional phenomics pipeline is usable, and that you might consider using it yourself. Thank you.
Well, thank you, Larry. So before we take questions, and there's a couple different ways we can do questions. Either you can put questions in the chat box for Larry, either address them to everybody or to myself or to Larry, and he can read them out. Or as we're unmuted here, you can unmute yourself and ask the question orally and just identify yourself beforehand. And Larry, I want to thank you. That was a very interesting presentation. So one of the questions that I sort of had is you did this interesting experiment where you cut roots. Is there some intention to go back and retrain or put those parameters into your simulation model then and try and update that simulation and improve it?
Yeah, that would be the idea of how they can work kind of hand in hand like that. In this case with maze, I sort of viewed this, I'm kind of rolling down in some sense, I guess, my maze work, but those were some outstanding questions in maze that I still had from my PhD. But we'll be doing some similar work in wheat, and we're trying to parameterize the wheat open stemroot version, and also look at the simulation model AppSim for doing wheat crop systems.
Okay. Do we have other questions right now?
I see there was one question of what the program was written in the chat, and the software we're working on here is mostly written in C++ with the OpenCV library and some API provided by the camera manufacturer. So we're just trying to release a compiled executable with all the libraries that will just work for anybody who wants to use it.
Okay. So I guess, unless you see any other questions, Larry, I guess, you bring up an interesting point. I'm assuming you're probably not trained or well-versed in programming in C++, and you did mention you were able to hire somebody in that, but really in this sort of transdisciplinary or interdisciplinary work in the space that we work in, how do you attract those people to your program so that you can, so you can really push the frontiers of this root imaging?
Well, yeah, so it's related to the field of computer vision. In my specific case, Anand, who's my computer vision specialist, was a master's student at University of Missouri with Alina Czar that Fritchie and I were collaborating with, and I had worked with Anand. And I think for them, it has to be an interesting problem from the computing side as well, not just a biological problem. So trying to figure out how to phrase your work in sort of challenging computer vision ways, I think, is necessary to attract people, and having good collaborators like Alina certainly helps.
Great. Any oral questions?
Don't be shy.
So if not, I'm gonna ask one more, and it's sort of a provocative or instigatory question, but so if we're interested in something like yield, right? Why do we care about measuring roots? Why can't we just select for better roots by measuring yield in enough fields?
Yeah, that's a good question, and one that I've thought a lot about. And this goes back to, I mentioned earlier, this sort of a tension or debate about IDEO type breeding versus selection for yield. or deficit elimination, like disease, And these are concepts that were contemplated by C.M. Donald from Australia, and he had come up with IDEO types for wheat. And to me, I would like to prove this or try to prove this somehow. But the way that I think it works is that, when you're selecting for yield, the plant is shuffling around its genetics, and based on that, also shuffling around its phenes. And when you're only selecting for yield, maybe you're selecting for the good states of some phenes, but maybe actually they're in a phenotypic background that isn't really up to snuff. And so, in other words, maybe the only way to combine 1,000 different phenes into one super plant will be to actually understand the operation of all those phenes and how they interact and to build them together. I think it's similar to, would you trust an airplane that was just built by randomly selecting for flight? That may actually work pretty well. In fact, people have used things like genetic algorithms to help design things like that. So it's sort of a similar concept as selecting for yield. But in the end, I really feel like if we have better understanding of how plants work and how to stack phenes, that should be able to overcome any barriers that selection for yield might have. And from a theoretical standpoint, I don't know how you really show that, but yeah, it's really worth thinking about.
Okay. I've not seen any questions in my chat box, but Larry, this is part of the interesting experiment we're always doing here is how do we attract people to show up for the webinar and to engage. And there was a few people who said, really, you're going to have a webinar during summer? And I said, for most of us, we still work during the summer.
Right.
But I've noticed that a lot of the grad students who normally participate in this did not show up today, so maybe they're all busy in the field working. Oh, we have a question. Okay, Brandon Hurst said, related to that question, your answer, how do we know which phene states are, quote unquote, optimal on an individual level?
Well, this goes back to the pipeline, and if you just kind of take a shotgun approach and measure all the phene states in a population and relate those to nitrogen uptake, shoot mass, even yield, that you can infer the optimal phene state based on the regression. So the optimal phene state is the one that creates the best plant per performance. And then, of course, it also feeds into the simulation models of what does the simulation show you. But yeah, I mean, it's not a clear-cut answer, and I know it's a lot of arm waving too, and that's kind of what the reviewers, I think, thought about my review as well. But I think that we really need to think about all these things and how the phenotypic background and how phenes interact. I really feel like we lack a lot of basic understanding in that area.
Yeah, so including the physiology in there.
Yeah.
Okay. We'll see no more questions. I'll just make my quick plug. I normally do this before we take questions, but for the Plant Phenome Journal, we were doing pretty good, getting some steady submissions, and I think right now I think we have maybe 2 papers that are in review, but I'd really encourage everybody, including Larry York, to consider the journal for some of this great work. I think it'd be a great opportunity for highlighting it. And I wish you the best of luck, Larry. I think you did a great job presenting today, and hopefully anybody can reach out to you. I know your email's pretty easy to find.
Yep.
They can reach out to you if you have any questions.
Sure. They can also, I don't know if you can still see my screen, they can follow Root Biology News on Twitter if they want to get more information about roots in general. I run this as a service to the community. It's not my personal Twitter account.
So I'm still fairly, I guess, new to Twitter, or haven't really figured out all the Twitterverse. So this is not your personal account. So so this is primarily retweets of other root?
Yeah, I mean, originally I was wanting to do like more original content and things, and in the end, I wasn't really able to meet that demand. So what Rubology News does is I view it as a service to the community that aggregates, like you said, tweets about roots, but it's curated by me. So if it were just a bot, for example, you'd be getting a lot of random stuff, probably stuff about teeth. If you start to look up things about roots on the internet, it'll bring up a lot of stuff from like dentistry. So this is a hand curated news source that, because I have 2,600 followers, I only search for content from those followers, and then I retweet that as it relates to roots.
Huh. Interesting. Well, thank you for your service.
About 10 minutes a week now.
Well, thank you for your service on that, Larry, and thanks again for an excellent presentation. And this concludes our month of June webinar, and the July webinar is to be decided, but hopefully we'll have an announcement in the very near future. So thank you, guys.
Thanks,
Thank you, Larry.